Welcome to Microbes Without Borders. We have a very special guest this morning. We have Dr. Keyhani with us. And Dr. Keyhani works on mushrooms. Actually not. He's a mycologist, and we have a chapter on mycology, and he's going to tell us a little bit more about it because I know very little about mycology. I know that there's some edible fungi and there's black mold in your house which causes trouble, and then some diseases. But I really don't know much. So we hope to get some enlightenment from Dr. Keyhani this morning about what mycology is and why he studies that subject. Dr. Keyhani, how are you?
Very good. Thank you for having me. We actually study the intersection between fungi and insects. And these are two of the broadest and most species-rich organisms on our planet. There are more different types of fungi and more different types of insects than anything else.
Yeah.
These organisms have evolved over millions of years to fulfill all kinds of environmental and ecological niches.
How did you get started being interested in mycology?
Well, it's a bit of a convoluted story. My background and PhD was in biochemistry and molecular biology, so I don't really consider myself a mycologist or an entomologist, someone who studies insects. But I had a broad enough background in fundamental biochemistry that I felt that I could study anything. And so when I came here, I was interested in organisms that can degrade different kinds of polymers and carbohydrates. And that sort of began this study of these fungi that we study, which are able to degrade the chitinous exoskeleton of insects. And chitin is composed of carbohydrates, and it's the second most abundant carbohydrate on our planet, next to cellulose and hemicellulose. And that initiated this sort of program of study in my lab. But since then, we've really gone beyond that and studied different aspects of these fungi and their interactions with insects.
So you've done that work for your PhD and then continued later on, or?
No, my PhD was studying marine bacteria. We actually studied marine bacteria and how these marine bacteria utilize resources in the ocean environment, including copepods, which are tiny little shrimp that are constantly, they're the sort of the feedstock and the basis for almost all life in the ocean. It's either something predates on them or they utilize the organisms in the oceans.
That's very interesting.
But I've come a long way since then into something very different.
So I've learned a little bit about your work this semester because my students are working a little bit with your fungus, and you're working on Beauveria bassiana, and it's an entomopathogenic fungus.
Yes.
That's a mouthful.
Yeah.
What does that mean?
And so entomopathogenic means an insect pathogen. This fungus is what's known as, so there are more than probably 1 million different fungal species out there, and maybe some 500,000 have been characterized. Another 500,000 await to be discovered and studied. Very few actually of these fungi have even really been studied. Of these, there's a small percentage that have evolved the ability to parasitize and infect insects. So we're talking about a small range of fungi that have this ability. And Beauveria bassiana is what's known as a broad host range insect pathogen. If you've got 6 to 8 legs, it'll make a go for you. But it's harmless against vertebrates, so if you have a spine, you're okay. And people have suggested their use for pest control. And this is since their early discovery in the mid-19th century until today, as a means of controlling agricultural pests, invasive pests, harmful vectors of diseases like mosquitoes, and all of these kind of types of insect pests. It's considered a natural organism, and it's compatible with organic farming practices and these kinds of things. And it's sustainable and environmentally renewable, and has this kind of applicability. Of course, chemical pesticides are what's most commonly used today, And we're studying, what my lab does is we study the process of what makes this small group of fungi pathogens of insects. What's unique about them? What factors do they have that other fungi don't have that allows them to target and kill insects?
That's interesting. So it's quite widely used in agriculture?
Well, there are a number of obstacles for practical application of these fungi. One is cost. Chemical pesticides are so much cheaper that for the most part these chemicals are used. I mean, these fungi are considered more environmentally friendly. But the second obstacle is that they're living organisms. So using living organisms for biological control or microbial biological control application is not easy. So there are all kinds of obstacles towards widespread adoption of using these organisms. There have been a number of successes in a number of instances. Specific strains have been used against locusts, and they seem to be effective. And in greenhouse environments where you can control conditions, they also seem to work well. But in large-scale agricultural applications, it's still limited.
So is it FDA approved? Is it allowed to use it?
FDA and EPA approved, EU approved, and they're commercially available. You can get them probably in some stores, or certainly organic kind of stores. Organic agricultural kind of stores and so forth. And the interest in these organisms sort of waxes and wanes over time. What's happened is that oftentimes a new inexpensive pesticide will come into the market and people sort of lose interest precisely because of the cost and handling issues. But currently there are very few new pesticides in the pipeline, and the costs of their application is increasing, and so they're becoming more competitive. My lab, we don't really study the applied aspects. We're more interested in the fundamental science of, we use it as a host-pathogen interaction to try to study these virulence factors and these kinds of interactions. It's also an important environmental and ecological interaction naturally occurring. When insect populations bloom, what happens? These fungal populations follow. So they follow sort of predator-prey kind of relationship almost in natural environments. And they're important epizootic. So that means it's an infection that occurs in nature of insects.
Okay. So you do population modeling of the changes in the populations as well? Is it a
We don't go into the environment much. We stay on the lab bench work. I mean, we have some interest in looking at these fungi with respect to ants and fire ants, because fire ants are an globally invasive insect species. Yeah. So we have done some studies in terms of that, but most of our work is kind of biochemical and molecular in nature.
Pretty cool. In some of your publications, you're talking about the arms race of different organisms in the environment. What does that mean exactly? What's an arms race?
So you can imagine that these fungi are pathogens and have evolved the ability to attack and ultimately kill their hosts. Well, the insect host does not like that, and so they fight back. And there's a selective pressure for them to fight back and to hold off any microbial infections. In fact, insects have very strong systems for inhibiting microbes. Most microbes it can handle very well, and it lives in a they live in a microbe-rich environment in the soils or whatever. So they have very formidable defenses. And in the example that we showed, so Beauveria is a broad host range pathogen, as I mentioned. It'll go after anything with 6 to 8 legs. But some insect species seem resistant to infection by this fungus. And in particular, some beetle species. And in particular, there's one beetle species which is known as the red flower beetle. It's actually an interesting insect because it has evolved and spread thanks to human agriculture. It lives essentially in an artificial human habitat, which is grain storage areas. And the species that have evolved over the past 15,000 years of human agriculture, 10 to 15,000 years of human agriculture, these guys can no longer live in the wild. They've adapted to this particular human niche of grain storage that they're no longer competitive in the wild. But in this grain storage, they're very good, and they're capable of proliferating and doing very well. They are, of course, undesirable, right? Because they spoil and eat the food the grain stores. And so there's a significant interest in control of these pests. And people have suggested and tried to use this fungus against these pests. But guess what? Of all the insects that are out there, this one particular beetle is highly resistant to infection by the fungus, Beauveria. Even though closely related species of beetles are susceptible. So this was sort of a puzzle that we were interested in solving. And what we demonstrated, or what we showed, was that the beetle makes particular secretions onto its cuticle, exoskeleton cuticle, and these secretions are very potent antimicrobial secretions. And so it's capable of killing or inhibiting the growth of microbes, including Beauveria. The Beauveria tries to fight back. It makes an enzyme that can detoxify these quinones. But what we showed is that the expression or the amount of enzyme it makes is insufficient to overcome the beetle defenses. If you artificially increase the expression of this enzyme, you increase the ability of the fungus to kill these beetles. But the fungus has no effect on other insects or that don't make these particular secretions. So we believe we have captured, in this instance, an example of an arms race between the insect and the fungus that helps explain the resistance of the beetle and an attempt by the fungus to fight back. So we think this is kind of like an evolutionary snapshot of what might be of what's happening, and at the present circumstances, the beetle is winning.
That's really interesting.
So that's one thing that we do.
That's very, very interesting.
Yeah.
Very cool. So of all the different mushrooms and fungi out there, if our students go on a scavenger hunt through the woods or through a house or a museum, what would they find? What kind of mushrooms, fungus can you encounter and just see with your naked eye?
Well, you need to know how to look. So there are the obvious ones, which are the mushrooms, which are the fruiting bodies, essentially, of the underlying fungi that are growing in the soils. So the mushrooms that you see are really these kind of sexual stages of fungi, and they're obvious. You can see it. Those are the kind that look like the mushrooms that we eat, and so forth. But underlying all of that, not all fungi form these kind of fruiting structures in mushrooms. So mushrooms is a particular subset of fungi produce these structures. But there's fungi all around us. And fungi have a number of unique properties. They're the great bioremediators. So they're responsible for turnover of biomass, the dead trees, decaying trees, and all of these kinds of biomass that's being turned over. Fungi account for a significant portion of it because they can target things that bacterial species often cannot, like the cellulose and the hemicellulose in the trees and these kinds of things. You will see anything that looks like it's decaying probably has some sort of fungal assemblage with it. Anything on man-made structures like on concrete, the black that you can see, or in your shower, that's all fungi. That's growing not in a mushroom form, but in a different kind of a form that you can see. So they're all around us. And aside from bioremediation, the second thing that they're very well known for is the production of secondary metabolites, including antibiotics, and the discovery of antibiotics, accidental discovery of antibiotics.
Can you share the story with our students?
Oh, I don't know if I know it very well. I know it's Ian Fleming, and he accidentally dropped some. So one of the most prevalent fungi, especially in human habitats, in the air vents and all of that, is Aspergillus species. And these species, there are some aspergilli that are human pathogens, but for the most part they're not. They can grow, on any kind of substrate. If I remember correctly the story, he had left a plate open, and an Aspergillus spore had landed on his bacterial lawn plate, and he noticed that the bacteria were not growing around this fungus that was growing. I suspect that's something that maybe other people had seen for a number of times, but what he made the important link is that, hey, this fungus is secreting something that's inhibiting the growth of the bacteria. And that kind of a very basic, simple assay is still used today to screen for organisms that make antimicrobial compounds. And so once he's made that connection between lack of growth of the microbe, of the bacterial microbe, to something being made by the fungus, then it just became but he had a pathway to trying to isolate that active compound.
Yeah, that's a fascinating story, how mistakes can lead to absolutely breakthroughs in science, and it happened quite a few times.
Yeah.
So with the antibiotics, isn't it sort of an oxymoron that soil organisms and fungi produce toxins? Is there a reason why they would produce these toxins?
Well, who are their main competitors? The main competitor of a microbe is another microbe. So in that sense, it would make sense that they make compounds to inhibit competitors. That also implies that they have mechanisms of resistance against the antibiotics that they make, which is the case in almost all instances. So I think from a competitive point of view, you gain an advantage by making these compounds.
That's another arms race in the soil.
An arms race in the soil, yeah.
Wherever we look at. So with the antibiotics, we ran a little bit into a problem with all the drug resistance and, not a big different choices of antibiotics in the pipeline. What do you think is going to happen in the future? Do you think we can solve the problem with antibiotic resistance?
Well, I'm optimistic. I think so. I mean, there's certainly a lot more that awaits to be discovered in terms of these secondary metabolites, and there are a lot of different approaches that people are using. There are a number of recent breakthroughs. A lot of fungi, for instance, that may have these bioactive metabolites, we don't know how to grow them or how to culture them. And there are new techniques that have been developed where you don't have to culture them in order to screen it. You use sort of a soil microbiota, an assemblage, and screen from that kind of a situation for new antibiotics. That's one approach. A second approach is to combine chemistry with this natural organisms and to go in and tinker with the genes to make different substitutions on old antibiotics so that they can increase their potency or mitigate any resistance that's been developed. So those are sort of two, I think, very promising approaches that are going to yield new compounds.
We've got a lot of work to do ahead of us.
Definitely.
So you talked about the secondary metabolites, aflatoxins and other toxins what fungi produce is another big problem. And I think often in food products, like peanut butter, oftentimes you can find some aflatoxin. Is that something that could potentially be something beneficial? We could learn something from used toxins?
Well, that's sort of the flip side of the antibiotics, right? Are the dangerous metabolites from a human toxicity point of view that fungi make. And they certainly make a lot of different kinds, including aflatoxin. And even in historical records, there St. Anthony's fire, which is also produced by fungi that infect grains, and led to people losing their limbs in the Middle Ages and things like that because they didn't know. I mean, they were eating this sort of fungally infected breads and so forth. What can be done about it? I'm not so sure what can exactly be done. I mean, we have a system where we screen, our food products for these kind of compounds, and we try to keep the bad fungi away.
It's always good and bad to the microbes.
There's good and bad to the microbes. And people sometimes try to use quote unquote good microbe to try to suppress populations of bad microbes. And that's one way.
So you teach in the department a class called Molecular Genetics of Prokaryotes.
Yeah, we probably should rename that because it's really now molecular genetics. We cover eukaryotes also. And yeah, I teach genetics. I mean, it's an advanced genetics class, and we go into the mechanisms of how transcription, translation, replication occur. We do some human genetics, and we also ask questions, broader questions sometimes students are interested in. How is the sort of explosion in human genetics information going to affect society and medicine at large? And we try to explore some of those questions also.
It's a very popular course from what I hear from the students.
I don't know about that.
So what gets you up in the morning to come to work? Why are you excited about what you do?
I can't envision doing anything else myself personally, because there's always something that awaits to be discovered, and for me there's no greater thrill than finding something or understanding something that nobody else has. Of course, it's a small piece of a bigger puzzle, but I'm happy with that. I like the idea of studying something that on its surface appears a little bit obscure, but underlying those mechanisms that we study, for instance, stress response in our fungi, those same mechanisms exist in man. We respond to stress on a biochemical and molecular level with some of the very same pathways in these small little fungi. And that's incredible, this kind of interchangeability and interconnectedness of life on our planet. And you study something here, and you may make a discovery that's important for a wide range of issues.
Yeah, that's very cool.
And there's always something.
Science is a lot of fun. You get paid for playing in the lab.
You get paid for yeah, exactly. And there's never an end to it.
That's true.
What we published last year, hey, that's old news. We got to move forward and build upon that, and build upon the ideas of others, and that's progress, I guess.
That's true. So my favorite fungi are the slime molds. I think they're fascinating because they can solve mazes, and they just have, they aggregate, although they're single cells. What is your favorite fungus?
My favorite fungus has got to be the one I'm studying.
That's true. That is true.
So Beauveria bassiana actually has an interesting history. It's named after an Italian scientist by the name of Agostino Bassi, and 19th century Italian scientist. And what happened was he was a contemporary of Pasteur, and he actually used Beauveria to develop the first germ theory of disease, maybe a little bit before Pasteur. And what he noticed was that in Italy at the time, they had a thriving silkworm industry. A few centuries before, they had stolen some silkworms from China, and they grew them, and they made the silk in Italy. Why get it from China if you can make it in Italy? And they had this silk industry. But periodically, the silkworm the silkworm farms would be devastated by a disease called white muscardine disease. And muscardine, I think, is a French word or Italian word for candy, because the larvae would look like little candies, white, mummified, fluffy candies. Certainly wouldn't want to eat them. And for us, when we see something like that, we say, oh, it's a fungus, clearly. But they didn't know that at the time. And what Augustino showed was that if you prick a dead insect with this fluffy growth with a needle, and you take that needle and you prick a live one, that live one will develop the disease. So it's really a demonstration of a germ theory of disease, and that was the beginning of this field, sort of fungal insect pathology.
That's fascinating.
One day I'd like to go. He was from Lodi in Italy. I've never been to Italy. One day I'd like to go to Lodi and visit his home place. That would be nice.
So have the course is called Microbes Without Borders. Through your career, have you been traveling quite a bit?
Yeah, there are a number of places where there are very active research efforts in this field. China is one. I have a lot of collaborations with Chinese scientists. Brazil is another place where it's probably one of the largest applied users of fungi for pest control happens in Brazil. And Turkey, all around.
So you've got to visit some of the places, and your colleagues?
Yes, definitely. Yes,
That's
I've been fortunate to have gone to China and Brazil and Turkey and other places where they use these fungi.
So being a scientist, not just the microbes travel, but scientists who travel a lot as well.
Yes, we do. It's a minor perk.
And one last question. I know from talking to you previously that you are interested in the interface between art and science. How do you think art and science go together?
Well, they share, the best science includes, I think, a creative process where you make connections between things that don't seem connected. And art, I think, can be the same way, where connections are made between themes that don't seem necessarily connected. And the end result is something that's new, a new experience. In science, of course, that experience is a description of our world, and hopefully, is some aspect of its reality. In art, it's a more subtle description of the same thing, on a more emotional level. And I don't see any, necessarily any a contrast between the two, or a conflict between the two. They can, some work, when you look at some electron micrographs of structures, biological structures, they really look like pieces of art. And vice versa, a lot of art is influenced by biology and the nature of the morphology of the world around us. So they're, I think, interconnected. Not all has to be. I mean, there's some art that's abstract and separate, and some science which is very esoteric.
And I think in an old days, some of the really great scientists like Da Vinci, a lot of them were artists and scientists at the same time.
Certainly. The ability to, draw was a very key thing. You see in many of the older texts, biological texts, they have fantastic drawings. They're all handmade drawings. We've lost a little bit of that. I think there used to be classes where you could take in doing that kind of stuff, medical drawing and things like that. And there may still be in some medical schools, but hopefully that won't be lost.
Do you have any other advice for our students? Anything what they can do, should do with their lives? Any advice?
Oh my goodness, that's a very difficult question. I can't remember. Let's see. I think that's the sort of the Kantian epistemology, right? He had 3 questions. What should you know? What can you know? And what should you do with what you know? So people have written whole philosophical texts on those 3 questions. My short answer for those is, what should you know and what can you know is as much as possible, I think. And what should you do with what you know? The simplest thing I could say is do no harm. So that's about summarizes.
And be excited about what you do.
Yeah, and be
find
excited
something
about what you
you're
do.
passionate about.
Yes, definitely, definitely.
Great. Well, we really appreciate that you joined us today.
It's my pleasure.
And if you want any more information about mycology or Dr. Keyhani's work, we'll provide you with information. You can find out more about him. Thank you very much.